Bismillahirrahmanirrahim. Assalamu alaikum. In this video, we are going to learn about the ketone body metabolism. Ketone bodies are the water-soluble compounds and they act as an alternate fuel for the cells during certain conditions such as the prolonged starvation, low blood glucose level, and the diabetes mellitus type 1 and in some ketogenic diets. Now, there are the three types of the ketone bodies that are produced in human body. These include the acetoacetate, the beta-hydroxybutyrate, and the acetone. Now, acetoacetate and the beta-hydroxybutyrate can be utilized in the body and they can be utilized in the brain and the muscles. Whereas the acetone is a volatile compound, it cannot be metabolized further in the body and therefore it exits via the lungs and is exhaled. One other point about the ketone bodies is that liver and RBCs cannot utilize the ketone bodies for energy purposes. Now we will see the ketone body metabolism, that is the process of the ketogenesis and the process of the ketolysis. Now, we already know that the first source of fuel in our body is the carbohydrate, and carbohydrate is in the form of the glucose in the cells, and the glucose is then converted into the pyruvate and then to the acetyl-CoA through a process called the glycolysis. And then after the glycolysis there is a process that is called the Krebs cycle or the TCA cycle, in which the acetyl-CoA through the various steps is converted into another product, that is the oxaloacetate. Now through this process various ATP, FADH and the NADH are produced during the Krebs cycle and they enter the electron transport chain for the purpose of the energy production. In certain conditions, when there is the low blood glucose level or there is the prolonged starvation or there is the low blood glucose level in the cell due to the decreased uptake as a result of the insulin deficiency in cases of the diabetes mellitus type 1, now this glucose level will decrease. As a result, the pyruvate level will also decrease. That will result in the decrease in the acetyl-CoA production. The production of all these products of the Krebs cycle will also be decreased. That will result in the decreased production of the energy, that is the ATP production through the electron transport chain. And when there will be the low energy source and when there will be the low energy in the cell due to the less carbs or due to the less glucose in the body, our body will use the fatty acids as the second source of energy, and the fatty acids through the beta oxidations will be converted into the acetyl-CoA. Now this acetyl-CoA level will be increased. This increased acetyl-CoA will enter the Krebs cycle and will result in the greater amount of production of the ATP. When there will be so much high amounts of acetyl-CoA, this will result in the increased amount of the oxaloacetate, and that increased fatty acid oxidation results in the decreased NAD plus over NADH ratio. So the increased amount of the NADH will shift the oxaloacetate to be converted into the malate. And this oxaloacetate will be diverted towards the formation of the glucose via the process of gluconeogenesis, which requires the oxaloacetate to come out from the mitochondria into the cytosol via the malate oxaloacetate shunt pathway, in which the oxaloacetate is first reduced into the malate by the mitochondrial malate dehydrogenase enzyme, and then this malate is reoxidized into the oxaloacetate via the cytosolic malate dehydrogenase enzyme. This oxaloacetate is then converted into the PEP, that is the phosphoenol pyruvate, via the enzyme phosphoenol pyruvate carboxykinase into the glucose. So this is all the process of the gluconeogenesis that is occurring by the beta oxidation of the fatty acid when there is the increased amounts of the oxaloacetate and when there is the need for the production of the glucose. When too much of this oxaloacetate will be used for the gluconeogenesis or for the production of the glucose, its amount will be decreased, and as a result it will not be able to bind to the acetyl-CoA to form the citrate. So the acetyl-CoA level will increase, and when there will be too much acetyl-CoA, it will be diverted to a pathway for the formation of the ketone bodies. So the first step in the formation of ketone body is the fusion of acetyl-CoA molecule with another acetyl-CoA molecule, and that will result in the release of the CoA, and in the presence of the acetyl-CoA acyl transferase enzyme, acetoacetyl-CoA will be produced. Then this acetoacetyl-CoA is converted into another compound, that is the HMG-CoA. This is the rate limiting step in which the acetoacetyl-CoA is converted into the HMG-CoA, and in this step there is the release of the CoA and there is an addition of the acetyl-CoA molecule in the presence of an enzyme, that is the HMG-CoA synthase. Then HMG-CoA is converted into the first of the ketone body, that is the acetoacetate, and in this process acetyl-CoA molecule is released, and this process is actually the lysis of the HMG-CoA. So the enzyme here used is the HMG-CoA ligase. Then this acetoacetate can undergo two pathways. It can be converted into the beta-hydroxybutyrate by an enzyme beta-hydroxybutyrate dehydrogenase. In this process the NADH is converted into the NAD+, or it can be converted into the acetone, that is the third ketone body. And in this process acetoacetate decarboxylase is used, So this is the process in which there is the decarboxylation. It was the 4 carbon compound, and now acetone is the 3 carbon compound. Through these processes, we came to know that beta-hydroxybutyrate, acetone, and the acetoacetate, all these ketone bodies are actually acidic in nature and have the pKa between the 4 to 5. Therefore, they decreases the pH of the blood and result in the acidosis, that is called the ketoacidosis. How this acidosis is diagnosed? It is diagnosed by the high anion gap due to the increased production of the ketone bodies, and one other sign of the acidosis in case of the increased ketone bodies is the cosmol breathing, which is characterized by the excessive, very deep and rapid inspiration. Acetone that is produced here cannot be metabolized further in the body, therefore it is exhaled in the air, whereas the other two ketone bodies, that are the acetoacetate and the beta-hydroxybutyrate, they enter the bloodstream. And one other point that should be noted here is that when they enter the bloodstream, blood circulation, they basically go to the brain, and where they come in contact with an area in the brainstem, that is the area postrema. Now this area postrema has no blood-brain barrier, therefore the substance, it can detect the substances in the blood, and it acts as a chemo trigger zone and will induce the vomiting. As a result, when there will be the increased acetoacetate or the beta-hydroxybutyrate ketone bodies in the blood, this area postrema will cause the vomiting, and this vomiting will then results in the hypovoleminemia, that is the decreased blood volume, and in severe conditions this decreased blood volume can result in the coma. So this was all the formation of the ketone body, that is the ketogenesis. Now we will see the ketolysis, that is the further metabolism of the ketone bodies. Now we know that these ketone bodies will enter their effector organs, or will enter the organs such as the brain or the muscles. In case of the muscles, this beta-hydroxybutyrate is converted into the acetoacetate in the presence of an enzyme, that is the beta-hydroxybutyrate dehydrogenase. And then this acetoacetate is converted into the acetyl-CoA in the presence of an enzyme thiophorase, or it is also called the succinyl-CoA ratio acetoacetate-CoA transferase. Now this acetoacetyl-CoA is converted or cleaved into the two molecules of the acetyl-CoA. So basically this is all the reversal of the ketogenesis, except an intermediate, that is the HMG-CoA, that was present here but it is not here. In place of the HMG-CoA intermediate, there is another compound, that is the succinate, which provides its CoA group and is converted into the succinate and results in the formation of the acetoacetyl-CoA. Now this acetoacetyl-CoA is cleaved into two molecules of acetyl-CoA in the presence of an enzyme, that is the acetyl-CoA acyl transferase. This acetyl-CoA and also this acetyl-CoA now will enter the Krebs cycle and will be used for the energy production that we already know. And so this was all the process by which the ketone bodies are used for an alternate fuel of the cell by producing this energy at the end. Now there is one clinical correlation that is related with the ketone body formation, that is the diabetes mellitus. In most cases of the diabetes mellitus type 1, there is the increased rate of the formation of ketone bodies and the reduced rate of use of the ketone bodies. That results in the increased level of the ketone bodies in blood, that is called the ketonemia, and in turn increased level of the ketone bodies in the urine, that is called the ketouria. Now this increased level in the blood results in the acidemia or the acidosis. Now this acidemia is because carboxyl group of the ketone body has a pKa of 4, that is the acidic, and also the ketone body loses its proton in the blood circulation. Therefore it results in the acidosis or the acidemia. The blood pH level decreases due to this. Now due to the urinary loss of the glucose and the ketone bodies, there is a severe type of the dehydration, and this dehydration results in the hypovolemia and a severe type. So this all condition which is a severe type of the acidosis is called the diabetic ketoacidosis. One other point to be noted is noted in case of the diabetes mellitus, as we know that due to the acetone cannot be metabolized further in the body and is exhaled in the air by the breathing. So there is the fruity odor in breath in the diabetes mellitus type 1 patient. The normal ketone body concentration in blood is about 3 mg per deciliter, and in case of the diabetes mellitus type 1, there is the 90 mg per deciliter in the blood, and in urine about 5000 mg per 24 hours ketone bodies are excreted. Another point to be noted is that ketoacidosis is not only seen in the diabetes mellitus, however it can also be seen in some other conditions, such as we already discussed. During the prolonged fasting, there is an increased formation of the ketone bodies, and due to the excessive ethanol consumption, there can also be an increased formation of the ketone bodies that result in the ketone acidosis.